By this time, you will have had health, grade 10, and various introductory bio classes, so you know that the functions of the reproductive systems are to produce gametes. Gametes are the sex cells. You have to move those gametes. You have to keep them alive. You have to be able to maintain the fertilized zygote and maintain a fetus for a woman when you're pregnant. And you also have to produce sex hormones, and males produce the majority of what they produce is testosterone, and females will produce estrogen and progesterone. We know that the genetic information is DNA. We know that in humans, each cell is diploid, which means that there are 23 pairs or a total of 46 chromosomes in each somatic cell. Gametes are those human sex cells, and they will be haploid. They will contain one set of chromosomes, only 23 of them. Meiosis, you should know the definition of this, is going to be the cell division that results in gametes. Each gamete possesses half the chromosomes that the parent cell has. So the male sperm is the male gamete, and that's going to have a single set of chromosomes, 23 of them. The ovum is going to be haploid and will also have 23 chromosomes, one set of them. In order to get back up to 46, you need two sets, 3 pairs, and the egg needs to be fertilized by the sperm. There are videos of meiosis all over YouTube. One that Khan Academy put out is fine. Anything that has a .edu in the title is fine. Or I should say, as part of the URL is fine. There are going to be 2 steps. So it's kind of like you go through mitosis twice. There are 2 steps. You have to duplicate all of the genetic material. Then you have to cut that in half by 2 cell divisions, which leaves you with haploid cells. In other words, cells that have 23 chromosomes each. One parent cell, which will have 23 pairs of duplicated chromosomes, turn into 2 daughter cells, which will have 23 pairs, 2 sets of duplicated chromosomes, into 4 gametes, which will have 23 chromosomes or 1 set of chromosomes. In spermatogenesis, which is meiosis in the male, it happens in the testes, and oogenesis, which is meiosis in the female, occurs in the ovaries. That's a great little video, which has a comparison of meiosis and mitosis. You're required to know that, so check that out. They are similar in that the DNA is going to be duplicated before the whole process starts. The difference, the major difference, is that in meiosis, there will be 2 consecutive cell divisions, conveniently known as meiosis 1 and meiosis 2. That shows you what's happening, gives you information about the number of chromosomes during each step. We will see that the stages are named similarly. So metaphase, prophase, metaphase, anaphase, telophase, except in meiosis, we'll have those happening twice. This is a great little chart here, so make sure you take a look at that. Interphase 1, chromosomes hanging out, and then they replicate. End up with 23 pairs of duplicated chromosomes. In meiosis 2, that is what's known as a reduction division. You have 4 stages. You have PMAT, first go around. Remember that you will also have cytokinesis. Cytokinesis is division of the cytoplasm. Let me repeat that. Prophase, metaphase, anaphase, telophase. 1, 1, 1, 1. All of these are the first step of meiosis. Cytokinesis is the division of the cytoplasm. Prophase 1 sounds familiar because the chromosomes are going to become short and thick. the nuclear envelope and the nucleoli both vanish. You're going to have the spindle fibers appearing, and chromosomes will form what are known as tetrads, which are these homologous pairs that are clumped together. In metaphase 1, meta means middle, those chromosome pairs are going to line up along the equator or the metaphase plate in the middle of the cell. Excuse me, in anaphase, the homologous pairs split. One of each pair is going to move to the opposite pole, and a cleavage furrow starts to form, which will divide the cytoplasm. Telophase 1 goes hand in hand with cytokinesis. They are not the same thing. Cleavage furrow is complete, which means that you have two different daughter cells, and each of those daughter cells will have half the chromosome number of each of their parent cell. Of their parent cell. So you'll have 23 duplicated chromosomes. Equatorial division is meiosis 2. You have stages that are similar to meiosis because the centromere will split between duplicated chromosomes, and sister chromosomes are going to end up moving to the opposite side of the cell during anaphase 2. Again, you have the same phases except the second go around of each, as well as cytokinesis. Result of this process is 4 gametes. They will have 23 chromosomes each if everything goes correctly. Result of spermatogenesis is 4 sperm. Let me repeat that. At the end of spermatogenesis, you'll have 4 viable sperm. At the end of oogogenesis, you have 1 ovum and 3 polar bodies, which are these smaller, non-viable cells because of unequal cytokinesis. And that shows you a chart of what's going on. So make sure you take a quick look at that. Now, there will be 2 videos that you're going to be watching on your own, which I usually show in class, which enforces viewing. One is the Great Sperm Race. It is required viewing. There are questions that come off that video that will be on the final exam. Your final exam, I should say the last module test, because you're going to panic and say, oh my gosh, we have a final exam. Is it cumulative? No, it's not. Make sure that you watch Great Sperm Race. It is rather entertaining, and it gives you a really good idea of how hard it is for a sperm to actually make the trip